Hello, this is Mrs. V. We are going to go through blood components. So hopefully you have printed off your coloring sheet. If you have not, stop this video now and you can find the link somewhere in D2L. So blood is a connective tissue, and if you remember from AP1, a connective tissue is more of a non-cellular matrix than it is cellular. We know that we can draw blood from a vein, and if we take that blood and then we spin it in a centrifuge, it is going to separate into its fluid-filled matrix, which is the plasma. So go ahead and write plasma up here in this block. Since plasma is that fluid portion, it is the least dense, so we're going to find it up here at the top. Plasma is a liquid, it is straw colored, it is about 5 times more thicker than water because of the components that are in plasma. There are proteins dissolved in plasma, so it makes it kind of thick and viscous, kind of like an egg white. It is mostly water, it's going to have electrolytes. Our nutrients are going to be carried in the plasma and distribute those nutrients throughout the body. Our gases, so oxygen and carbon dioxide, and like I said, these proteins. We're going to learn about them. So albumin is a protein that is going to basically give blood its viscosity and also osmotic balance. If you remember, osmosis is the movement of water. The cardiovascular system's job is delivery, and we'll be delivering things like oxygen and nutrients to the tissues. And then we're going to be picking up carbon dioxide waste and other nitrogenous waves. What we want is an isotonic relationship between the liquid, the plasma in the blood, and those cells. So albumins are going to do that for us. Our second type of protein are globulins, and that word just means that these are spherical proteins. Some things cannot go into the watery plasma and they require a carrier. From chapter 1, remember we learned that hydrophobic or steroid or fats cannot be carried, so think of those hormones like thyroid hormone, they require a carrier in the blood. Other globulins are our immunoglobulins or antibodies. We've heard a whole lot about those protective immune proteins. They're also going to be in the plasma. Fibrinogens, these are going to be clotting proteins that are circulating in an inactive state, and blood clotting is called hemostasis, and so that's going to prevent us bleeding to death. And then this is also where those hormones, so regulatory proteins like hormones, will also be found in the plasma. So this is going to be the fluid matrix part. Below that, because they're going to be more dense than the plasma, we're going to get our formed elements. And the next thing, this little white disc right here, don't color it, just go ahead and leave it white. This thin layer of cells is called a buffy coat. This is what I used to do research on, and I would go to the blood banks and I would collect the buffy coat, because in the buffy coat, that is where platelets, which are involved in blood clotting, we'll color those in a little bit, and the white blood cells, also called leukocytes, that's where those are located in that thin little layer. So less than 1% of blood is going to be comprised of white blood cells and platelets. By far the heaviest cellular components are going to be the red blood cells. So go ahead and color this red if you have it. If you don't have red, use whatever color you want. But that is where the red blood cells or erythrocytes are located. So blood is 55% plasma, less than 1% of that buffy coat, so white blood cells and platelets, and then 45% red blood cells or erythrocytes. This is a blood smear, and all of these sort of pink small spherical cells, these are red blood cells. Erythro means red, cyte means cell. These are the smallest of the formed elements. They are small and flexible. They are basically empty membranes, if you want to think about them that way, in the sense that they don't have a nucleus, they don't have mitochondria, they don't have cellular components. About 97% of that cell is going to be hemoglobin. The job of the red blood cells is to transport oxygen, which we need to make ATP, the cellular currency of energy for all of our cells, and it can also carry some carbon dioxide or waste gas. If you notice from the top, like I said, they look like donuts that you didn't kind of punch the hole out properly. If you look at them from the side right here, you notice that they are hourglass shaped. So you want to go ahead and color that in. What this does is it gives a high surface to volume ratio. Oxygen is just going to diffuse from greater pressure to lesser pressure. So we pick up oxygen in the lungs. And then when the blood goes through the smallest blood vessels called capillaries, then it delivers that oxygen to the tissue so they can make ATP and do their cellular work. And then carbon dioxide is our waste gas. They are small, they're about 7 micrometers. And to go through our smallest vessels, they can kind of stack up like this. This is called a rouleau. And that's how they make it through the smallest of our blood vessels. Fitting through these smaller vessels, these cells, they kind of get beaten up. They don't have a nucleus, they can't reproduce on their own, they can't repair, so they only live about 120 days. When they start to get kind of beat up and ragged, when the blood is filtered in the liver and in the spleen, that's when larger white blood cells, macrophages, are basically just going to eat them. It's like, hey, thank you for serving for 120 days. We're going to eat you now. You are now going to be retired. If a person does not have enough red blood cells or has too little hemoglobin, it leads to anemia. You've probably heard of anemia. So if someone is anemic, they will have low energy. They may get winded when they move around, and they may appear pale. But the number one job of the resource sites is to transport oxygen. And for the most part, we're just going to use 45% of blood is going to be made up of the red blood cells. And we'll talk a little bit more about that in lecture. Our next cell is going to be the precursor, or the mother or father, of our platelets or our thrombocytes. And so this next cell we're going to talk about are called mega, because they're mega big. Karyo for nucleus sites. And so this is a megakaryocyte. I gave you this picture. They are an unusual hematopoietic cell. They are going to be in the red bone marrow. Notice they are large, and this is kind of showing you the progression of a megakaryocyte growing. And what they do is they hang out in the red bone marrow and they make these little cytoplasmic extensions called proplatelets. And then they just pinch off little bits of themselves. And so these are platelets. And inside of those platelets are going to be chemicals that are necessary to form a platelet plug, which is going to kick off blood clotting, which is called hemostasis. So thrombocytes is the name for platelets. A thrombus is a blood clot, a cyte is a cell. When we look at a blood smear, we learn that these are the red blood cells. These little tiny bits of purple, they almost look kind of like trash on a blood smear. Those are platelets. So platelets come from megakaryocytes. And all hematopoiesis occurs in the red bone marrow. Hemostasis, which you'll also learn about in lecture or from your textbook, is the stoppage of bleeding. The platelets, what they're going to do is they're going to stick to the walls of a broken blood vessel and make a temporary platelet plug. And then through a blood clotting cascade, then our body basically manufactures these fibrin strands. So this little green in this picture. And so the fibrin strands with the platelets and some trapped red blood cells, that's what you're looking at when you look at a scab. And so that is a true blood clot. Okay, so hopefully you have that one side of your picture done. This other side, we are now looking at leukocytes. So leuko means white, cyte means cell, or we can abbreviate that WBCs, white blood cells. So leukocytes, they would actually be clear. So to see them, we actually have to stain them. And so we would take blood, we would make a blood smear, and then we would use something called a Wright stain. The Wright stain, they used two different stains. They used a basic stain and an acidic stain. And back in the day in the 1800s, they looked through their microscopes and they realized that some of the cells you could see grains or granules. So they grouped those together and called them the granulocytes. So the granulocytes. The ones that didn't have granules or they didn't look grainy, they called agranulocytes, so without granules. And we have 5 different types of white blood cells. And remember, when we look at this centrifuge tube of blood, we have the plasma, which is the fluid matrix. We had our red blood cells, the erythrocytes carrying oxygen, and then this thin layer, so less than 1% of the cells, are going to make up the white blood cells, and we already discussed the platelets. So the first cell we're going to talk about are the neutrophils. They gave them the name neutrophil because they accepted both the basic stain, which was a blue, and the acidic stain, which was pink, and so these cells stain purple. So neutro, like neutral, and then phil means to love. So this is what it looks like. So in lab, you'll be looking at some blood smears, and they're really easy to identify these. Notice this is supposed to be the nucleus. It has a multi-lobed nucleus, so that's kind of the tell. It doesn't look like any of the other white blood cells. White blood cells are about 2 times as large as a red blood cell. And the job of the neutrophil, so they have these dark multi-lobed nuclei, and then they have these granules which stained purple as well. These granules are going to be used when these white blood cells defend our body against infection. So it's part of our immune system. That is the job of the white blood cells. They are the most numerous of all the white blood cells, and when there is an area of infection or injury, so out here in the tissues, they receive signals, and so they leave the bloodstream and they go to that area of infection. And they are highly phagocytic, and remember phagocytosis, that's cell eating, so they eat pathogens, so anything that could cause disease to us, so bacteria, fungi, virus particles. These granules that they have, there are their weapons, and so they're really effective against bacterial infections. And so when we do a differential blood cell count, we'll see that the number of neutrophils, the white blood cells, goes up when we've been exposed to a pathogen or have an infection. They are clearly the most numerous type of white blood cell when you look at a blood smear. So you won't have any trouble finding a neutrophil when you look at a blood smear. Our next type of white blood cell, they are called eosinophils because they loved the stain eosin. The eosin stain is pink, and so these are visually distinctive. Pretty much all the nuclei are going to stain purple. They have a bilobed nucleus, kind of looks like a pair of sunglasses or earmuffs or headphones, old-timey headphones. And then the granules that they have different weapons inside them, and the granules stain pink. So think of eosin, eosinophils, they're going to look more pink than any of the other white blood cells. Their granules make them very good at killing and neutralizing helminth or worm infections, so parasitic worms, so think tapeworms, flukes, roundworms. They are capable of phagocytosis, but then worms are kind of large, and so when a worm has been tagged, these are antibodies, then the eosinophils, they're all going to gang up on this worm and release their granules that are basically going to digest and kill this worm. They also have a very complex role in allergies and autoimmune disease. They can also get involved and do damage to our own tissues. So eosinophils. Neutrophils, purple nuclei, purple granules. Eosinophils have the pink granules. And then our third group of granulocytes are called basophils. So basophils. They accept the basic stain. When you hear basophil, I want you to equate them with our response of inflammation. So redness, heat, pain, swelling, itchy, watery eyes. And that basic stain was a dark blue, so I went ahead and colored these large granules a dark blue color. This is what they look like in a blood smear. The granules are larger than the eosinophils and the neutrophils, and so they almost just completely cover up the nucleus. It's kind of look like a blueberry or blackberry. And these cells, they are the rarest of all the white blood cells. I'll show you a blood smear in a second. But what basophils do when there is an area of injury or infection, they are going to respond and they release chemicals that are going to enhance inflammation. So think, I said, redness, heat, pain, swelling. We don't like inflammation, but it basically calls our immune system to that area of infection or damage and sets the stage for repair. How it's going to enhance inflammation, it's going to release heparin, which is an anticoagulant, which is going to allow blood flow and movement to that area. Histamine, which also increases capillary permeability. So think leakiness. This is why we get swelling in areas of infection or injury. And then eicosanoids, these are communicating molecules that are derived from arachidonic acid. So leukotrienes, they too are going to enhance inflammation. And so this is part of our innate immune response to injury and to infection. So this is a blood smear. If we were having class face-to-face, I offer a prize for anyone who can find a basophil. They are the rarest type of white blood cell. And so on this smear, you see a whole lot of red blood cells. They carry oxygen. You see these little tiny purple dots, which are the thrombocytes of the platelets. And then all of these are neutrophils. So they're the most numerous of the white blood cell. They're our first responders. Okay, so we did all the granulocytes. So the last two remaining white blood cells are called agranulocytes because you don't see large granules in the cytoplasm. The first one we're going to talk about are called monocytes. So one cell, but they are the largest of the white blood cells and they have only one nucleus, so monocyte. Go ahead and color the nucleus. It's a kidney bean shaped nucleus. And then it is surrounded by sort of lilac or light purple color cytoplasm. The monocytes are the largest of any of the cellular components in the blood. So they are easily two red blood cells across, maybe even a little bit bigger. So they're pretty visually distinctive. When these cells leave the bloodstream and they enter the tissues, they become macrophages or macrophages. Macro means large. Phagocytosis was eating. These are our white blood cells. They eat cellular debris. They eat pathogens, so fungal spores, bacteria, virus particles. These cells are longer lived than the neutrophils. They can eat over and over and over again. And so you'll see these elevated if somebody has a chronic infection. So acute infections are very quick, chronic infections are slow to develop and long lasting. And so if someone has an elevated monocyte count in their blood, that means their immune system has been fighting for a long time, and therefore the body's responding by making more monocytes. So you can also see this count go up with autoimmune diseases. Our last white blood cell that we're going to talk about are called lymphocytes. So lymphocytes, these are going to be the smallest of the white blood cells. These are the cells that are responsible for our adaptive immunity. So the part of our immune system that gets better over time and leaves us or makes us immune to infection. Go ahead and color the nucleus. They're mostly nucleus, so a very round spherical nucleus surrounded by just a little bit or a thin layer of lilac cytoplasm. There are two types of lymphocytes, and if you've been around or watched the news at all in the last 2 years, you've heard a whole lot about T cells, you've heard a whole lot about antibodies. And so T cells are our cells that respond to virally infected cells or cancer cells, and so they kill those cells. And then B lymphocytes become plasma cells that make antibodies. And we've heard a whole lot about antibodies and how antibodies are immune proteins that protect us against disease. Okay, so hopefully you got all that. Once your picture is complete, take a picture of it and convert it to a PDF and upload it to the Dropbox. As always, if you have any questions, feel free to reach out to me through Remind or through email and just ask. Thanks. Have a great day.